Neuroplasticity, arguably the most important understanding and principle of the brain, is what makes this living organ quite a challenge. Its ability to adapt and continuously change in response to genetic programming in the environment is amazing. In this chapter, we will focus on the incredible process of neurodevelopment, which began with the egg being fertilized by the sperm, and how this began its journey to becoming the adult brain that you have. This chapter has three general ideas: the amazing nature of neurodevelopment, 2, the important role of experience in neurodevelopment, and 3, the dire consequences of neurodevelopmental errors. This module will focus on the 5 phases of neurodevelopment, starting with the fertilization of the egg and how the cells become totipotent until they become unipotent, which is when they reach the ability to only be one specific type of cell. The journey to become who you are today started off with the fertilization of an egg. This fertilization eventually started becoming specialized and began to take form that eventually looked like this, the adult brain. Please remember that because of the nervous system's plasticity, we understand that the brain can actually change as a result of not only your genetic predisposition, which is what you would have inherited from your parents, but also with the environmental circumstances. So individuals that have been exposed to some circumstances that are not ideal would have a slightly different brain than individuals who have been exposed to an ideal environment. And we've seen this time and time again through research with animals, as well as studies that have been done with individuals' prison systems. We also understand that when things go wrong with our development, there can be some severe consequences. In this chapter, we'll be looking at some examples of some of those things. Case study of Jeannie, this child that at the age of 13 was rescued from her father and terrible living conditions, is a child who illustrates the impact of severe deprivation during development and how this impacted her permanently for the rest of her life. Jeannie, at the age of 13, was rescued by social services, and they found out that since the age of 18 months she had been confined to a room where she had been beaten, she had been starved, restrained, and it kept in dark conditions, for the most part just denying her of normal human interactions. When rescued, even with special care and training, her behavior never became normal. Jeannie could not stand erect, chew solid food, or control her bladder or bowels. Since the age of 18 months, Jeannie had spent most days tied to a potty in a small, dark, closed room. Her only clothing was a cloth harness, which kept her from moving anything other than her feet and hands. In the evening, Jeannie was transferred to a covered crib and a straitjacket. Her father was intolerant of noise, and he beat Jeannie if she made any sound whatsoever. According to her mother, who was almost totally blind, Jeannie's father and brother rarely spoke to Jeannie, although they sometimes barked at her like dogs. The mother was permitted only a few minutes with Jeannie each day, during which time she fed Jeannie cereal or baby food. Jeannie was allowed no solid food. Jeannie's severe childhood deprivation left her seriously scarred. When she was admitted to the hospital, she had almost no sounds and was totally incapable of speech. After Jeannie's discovery, a major effort was made to get her development back on track and to document her problems and improvements. However, after a few years, Jeannie disappeared in a series of legal proceedings, foster homes, and institutions. Jeannie received special care and training after her rescue, but her behavior just never became normal. into terms of humans. The following were a few of her continuing problems. She did not react to extremes of warmth and cold. She tended to have silent tantrums during which she would flail, spit, scratch, urinate, and rub her own snot on herself. She was easily terrified, specifically of dogs and men wearing khaki pants. She could not chew. She could speak only short, poorly pronounced phrases. Jeannie is a perfect example of how the environment could be so detrimental for the development of the brain. Jeannie is currently living in a home for retarded adults. Clearly, experience played a major role in processes of Jeannie's neurodevelopment. We'll look at the rest of the chapter and look at these processes. Please note that the term retarded adults is not in any way derogative or offensive. Retarded is actually a term that is adequately used with individuals that have a delay, hence the root word in there, tardy. That basically means that there is a delay in the maturity of individuals. I know that in some places and for some individuals this could be rather offensive, but please note that there are actually organizations of individuals who are trying to claim this word and make it an appropriate one as long as it's used for correct purposes. When individuals use it to belittle other individuals, that's when this word is taken out of context and it becomes offensive. But for the purpose of this, please note that it is not in any way derogatory or offensive. It simply is to illustrate and describe individuals who had delayed in their development. At conception, when the sperm meets the egg, the amalgamation of the ovum and the sperm becomes known as a zygote. This is a single cell that was formed, again, from the sperm and the egg. The zygote eventually divides into to form two different daughter cells, and these two will divide into four, and the four will divide into eight, and so on. Eventually, a mature organism is produced. But there's a lot more to development than just this. Had we just continued this division, we would end up with some form of amorphous mass, meaning there's some type of formless and no specific shape. For proper organism development, we understand that cells must differentiate. That is to say that cells become muscle cells, and they become neurons, and they become heart cells and lung cells, and through this specialization, cells begin to shape us into what we are today. The cells must, in addition to producing, they must also migrate and make their way to appropriate sites so they could eventually align themselves with other cells around them, and they would begin to form particular structures. I want you to think of tissue, and I want you to think of every body part that you can imagine. Once they migrate to this specific place and align themselves, they must also establish appropriate functional relationships with other cells. Not doing so can actually compromise the development of that tissue and can bring about deformities. The organism that was conceived remains as a zygote for the first 2 weeks after conception. From weeks 2 to 8, it becomes an embryo. During the embryonic stage, new cells become more and more specialized. Eventually, these cells can develop into different cells of only one class. For example, you may have different kinds of blood cells, different types of neurons. These new cells are said to be multipotent. But most developing cells will eventually become unipotent, and that is that they can only develop into only one type of cell. This is what we understand about stem cell research. Though controversial for some, as it requires the use of humans so that we can understand human difficulties, we understand that stem cell gives us an advantage over understanding things a little bit better. The understanding of the development of the cells gives us the ability to shape these cells into whatever cell we'd like them to be. Remember that as we are advancing in our development, these cells lose their malleability, if you will, and they begin to be very focused and very committed to the type of cell that they will be. But at the totipotent stage, This is when cells have the ability to develop into any class of cell in the body. This malleability is what allows the cell to become any type of cell, a bone cell, a skin cell, a tongue cell, a heart cell, a neuron cell. And this totipotency, which is what makes a cell very malleable, is lost just after 4 days of the embryological development. Please keep in mind this is in weeks 2 to 8. At this point, the cells begin to specialize, and with this specialization, they begin to be more committed into the type of cell that they will be for the rest of their life. The pluripotent stage is when cells have the ability to develop into many, but not all, classes of body cells in the body. So you could see the cells becoming more specialized as the embryo develops and eventually reaching the multipotent stage, which allows the cells to develop into different cells, but of only one class. For example, you could have a neuron cell that at some point could have developed into a heart cell or a bone cell, but now that it's become very specialized, it can still become different types of neurons, interneurons, bipolar neurons, multipolar neurons, but it cannot be anything other than a neuron cell. And eventually reaching the unipotent stage, and that is where they can develop into only one type of cell, and that would be, in this case, for example, bipolar neurons. It can no longer deviate into multipolar, it cannot deviate into anything else. It is now committed to that because of its specialization. Please note that what was just developed about the embryological development and the cells is occurring at a time where some women are not even aware of the fact that they're pregnant. This would be between the weeks 0 to 8, so the first 2 months of pregnancy. The developing neurons eventually are aiming toward specialization, and this is achieved in 5 stages. First, we're looking at the induction of the neural plate, which will eventually become our brain, a patch of tissue, which would be in the dorsal surface of the embryo. The neural proliferation, and that is the addition of neurons that are added to the specific area. The migration and aggregation, which is the cell making their way to the specific part where they need to then bind to other cells. Axon growth and synapse formation, which we'll talk about in future modules. And also, very importantly, the death of neurons and the synapse rearrangement. In order to understand our humble beginnings, we have to look at the neural plate. This patch of tissue that is found at the dorsal surface of the embryo eventually will become the human nervous system. This is visible at 3 weeks after conception, so please know that we're talking about the embryonic stage. When we look at the embryonic stage here, you're looking at the representation of the dorsal surface of the embryo. And if we do a cross-section of the dorsal, this is what this would look like. This green and purple part right here is what would be called the neural plate. Please note that the neural plate cells are often referred to as embryonic stem cells. And the stem cells basically meet 2 specific criteria. One, they have almost unlimited capacity for self-renewal if they're maintained in appropriate cell culture. And 2, they are pluripotent, and that is that they can become any kind of mature cell. Totipotent, again, is the earliest cells that have the ability to become any type of body cell. Multipotent are those that with development neural plates, our cells are limited to becoming one of the range of mature nervous system cells. But please note that for the purpose of illustrating what cells we're looking at here, the stem cells would actually be pluripotent. Tissue that's been taken from the dorsal mesoderm of one embryo. We're talking about the one of the development stages in here. And they're implanted beneath the ventral ectoderm of another embryo, and that would be like the host, induces the development of an extraneural plate on the ventral surface of the host. That is to say that by doing research on this in here, we could develop cells that would be needed for individuals that may be lacking them. For example, if there's a degenerative disorder of an individual whose brain is just simply not responding well and not developing like it should, we can, by making use of cells at the neural plate, develop these type of cells, the neurons that are needed, and then ideally in the future be able to administer this in here and induct them so that the nervous system could continue its course, either naturally or with the aid of processes and techniques. Please note that there are 3 layers of the embryonic cells. We have the ectoderm, that would be the outermost, the mesoderm, which would be the middle part, and that's what we looked at right here, the development of induced signals from the mesoderm. The endoderm, which would be the innermost, and we find that by 40 days after conception, the swellings are visible in the anterior end of the human neural tube. So this would eventually become, it went from looking flat like this to eventually just closing in and eventually looking like this. These swellings ultimately develop into the forebrain, midbrain, and hindbrain. Here you have a recap of the 5 stages. We started off by fertilizing an egg, and they start off as totipotent cells. Here's a description of totipotent cells, which are those that have the ability to become any type of cell in the body. Eventually they begin to be more specialized and they become pluripotent cells. They can still become different cells, but not all types of cells. When they reach the multipotent level, that means that they can only develop into different cells of one class. And then the unipotent cells would be those that can only develop into one specific type of cell.